range extensions, and different represented fauna in the area. We're also going to be looking for geological samples. I know Val is really ready to find some cool rocks, and they can tell us a little bit about basically the history of our planet, the history of life on our planet, and the history of the kind of geophysical processes on our planet. During this dive, we are also going to be collecting some eDNA, or environmental DNA, and we actually have two different methods for doing that. The traditional method that we have been using in the past for several years, we'll be collecting our Niskin bottle water samples, and then we also have a really cool new technology on board, the Multipuffer sampling system, which allows us to collect eDNA samples over a large period of time as we descend through the water column and then explore the seafloor. So send us your questions. And again, my name is Matt Holden. I am the Science Communication Fellow for this 12 to 4 watch schedule. When I'm not out at sea, I am a high school science teacher in Fayetteville, Arkansas, mainly teaching biology. So super excited to have the opportunity to be out here with all these amazing individuals, learn from them, and see what we can discover.
There's a little anemone. We saw a bigger version of that one earlier. Oh, a little sea star underneath that.
And we can go ahead and start with some rounds of introductions, especially for those of you who are just tuning in, or maybe this is your first time checking out the live feed on nautiluslive.org. So we have several people in the control van right now helping with this dive and helping us explore the ocean floor. Annette, would you mind giving us a short little intro of your background?
Sure. Hi, everybody. I'm Annette Govindarajan. I'm a science co-lead.
Thank you.
And I'm a biologist at the Woods Hole Oceanographic Institution, and I develop and apply eDNA methods to study deep sea biodiversity, looking at how animals are distributed and and how those distributions are changing with time. So
that's amazing. I love that. Looking really to see how changes in life down here on the seafloor may change at temporal scales and geographic scales. Super, super interesting.
Yeah, thanks. Yeah, so we have the new Multipuffer large volume sampler on Hercules right now, and so we've applied this and the similar samplers to water column platforms like the robot Mesobot, and done that very successfully. And now we're moving deeper and seeing what we can discover here.
Speaking of nav, could I get a pin dropped for Multipuffer start? It was back like 2 minutes. Sorry, I didn't get in the moment. And then these will be pumps 2 and 3, and the sample numbers are 078 and 079. Thank you.
Let me take a look at this coral here.
Yeah.
We saw this a bit deeper down the slope, but it's good to just check in, make sure we still know what we're looking at.
Yeah, definitely. That other voice that you're hearing right now is one of our other co-lead scientists. Steve, would you mind introducing yourself a little bit?
Sure. My name is Steve Oskovich. I'm one of the biology co-leads on this expedition. I am a deep sea biologist specializing in seamount environments and deep water corals, of which we're looking at one right now.
Yeah. Oh, these corals are just so cool every time I see them.
Yep.
Oh, it looks like maybe something's hanging out inside there. I thought I
Yeah.
saw something move.
There's almost always a squat lobster associate with these golden corals. This is a golden coral in the family
Go, buddy. Ready for 20 more
Chrysogorgiidae.
meters, you think?
And there's
Yeah,
a
I'm
particular
ready.
genus
Okay.
that likes
Yeah, let's do
to
it.
inhabit these. But this is actually a
Good.
coral called parachrysogorgia.
Parachrysogorgia.
Yep.
Cool name.
And this particular species has been sampled quite widely. It's very widespread. We can probably put it to one of two species, both of which have been sampled widely, but a lot of the times it's very difficult to discern species from
Happy. Thank you.
imagery, but oftentimes if we have a collection, we can better detail or put it to a species name if we have lots of other imagery and collections. So
yeah, so that's one of the important reasons that we are not just looking at these corals, but we're also collecting some specimens so that we can have further processing and analysis and really kind of pin down the exact species we're looking at.
yeah, this is one of the more common ones you see across the seamount landscape at these depths. These golden corals are a family that's exclusively deep sea for the most part. Sometimes you can see them up in shallow as the mesophotic zone.
Awesome. And why are they called golden corals? Is that because their skeleton is kind of goldish color?
Yeah, they have, if you were to scrape away some of the tissue and some of the polyps, they have a golden luster.
Yeah, I think I saw one we collected on an earlier dive down in the lab. It's just gorgeous. It's almost like reflective. It's so gold.
Check out our balsama, maybe.
We can go ahead and continue some introductions. Mariana, would you mind giving us a short introduction of yourself?
Oh, this one.
Hi guys, my name is Mariana Poznansky. I'm from the Marshall Islands and I'm with the science team sitting at the Science 2 seat here in the control van. And when I'm not at sea, I'm a student at University of Hawaii at Hilo, and I'm very grateful to be here with you guys.
Yeah.
And yeah.
Sorry, I didn't mean to interrupt you. I was just going to say, yeah, we're so excited to have you on board. Where I'm from in Arkansas, Mariana and I were having a discussion. It's such a small world because she's actually visited there.
Oh yeah.
And she has family that lives, within 5 miles of where I live in Arkansas, which is really, really interesting and really amazing.
Small world, huh?
Yeah.
I didn't know that. That's so cool.
Yeah.
Super cool. I told her next time she comes, she has to let me know so she can visit and I can show her around. Leela, would you mind introducing yourself a little bit?
I can indeed. Hi, y'all. I am Leela Madhava. I am the data logger for this shift, which means I'm noting down important events and keeping track of samples as they happen. I also help out with some of the sample processing after dives. I'm originally from Chicago, but I now go to school at University of Washington, Seattle, where I am studying marine biology and biology.
That's awesome. I love that. Super exciting to have you on board. I also studied biology in college. I'm sure many people, especially in our science team, did as well. So the more biologists, the better. We can all nerd out together.
For sure.
Video and zoom in on this one.
Copies of it.
We'll frame it up.
Oh, wow.
Yeah, we're looking at a glass sponge here. This one looks like it's in the family Farreidae.
What are some kind of distinguishing characteristics, Steve?
Mostly overall morphology.
Okay.
But the Farreidae are typically, have a large kind of voluminous three-dimensionality.
Okay.
But this, I'm not sure too about the problem with these is that they're so fragile to collect, and oftentimes if you try to collect them, regardless of how you collect them, they shatter into a bajillion pieces.
Oh no.
But this is a good shot. Let's just
Yeah.
hold that shot and then carry on.
It's really cool. I love all the openings and pores, and so that's how they'll.
There's a polychaete worm here associate too. I was saying on the last one I saw that too.
Oh yeah.
It's a tiny worm. They live in and around the sponge. They might feed on other, bits of food.
Amazing.
That
A little more zoom. Like try and get on that.
Oh, that's great. Wow. I didn't even see that until you pointed it out. Again, Steve, you have like eagle eye vision with these species. It's incredible.
Mm-hmm. Nice.
Nice. Thank you.
Yeah.
Looks like I got a little more tether.
And again, those two little green dots are lasers that we have on ROV Hercules, and they are 10 centimeters apart. So that gives us a good idea of how large these organisms are that we're observing.
We saw that sponge a bit deeper down the slope too, so
Okay.
it has we can start to put some depth constraints on its distribution.
Yeah.
So
That's great.
we haven't moved much up slopes, and even since when we started the dive earlier, in our previous dive, waypoint 1-ish, we moved up about 100 meters-ish, maybe 150. So there's more to come. The seamount rises quite quickly
20 more meters in, right?
over the next
excited.
several hundred meters.
Yeah.
So just to
Awesome.
confirm, this is now
Yeah,
H2112?
It's not right.
Copy.
Yeah? Okay, cool.
Bridge nav.
Let's see here.
Can you... Noelle, I didn't see her when I came up. Can you hear me? Okay. Yeah, I didn't see her when I came up. Do you know who his roommate is? Okay. Let's see. I'm trying to think who was up that's not on watch. Chris is up. Yeah. Uh-huh.
Oh, what's that? It's not a slug.
Would now be a good time for the ROV pilots to give us a short introduction?
There might be something right here. Do you see that stick? That's kind of...
Yeah, I'll go up to the stick.
Yeah. Oh, I see you're going for the
I was going
cucumber.
for the golden ball on the bottom, and then I was going to go for the thing on the...
Golden ball.
Or the...
I don't see it.
Am
What's
I
this?
blind?
Yeah, what's this ball down here? Zoom in.
Oh, I see a sea
Zoom in.
cucumber,
Oh, I
think.
see. Yeah. Oh, yeah. I think we're looking at the cross-sec. Actually.
Oh,
Good eyes, Levi.
yeah. Great eyes.
I just don't know what it is.
I don't know what that is either.
Maybe it's a ballast rock from a...
Exciting. I love when we find things that especially Steve doesn't know.
Well, I mean, usually it's not obvious.
Is it an anemone?
I don't think so, but it could be. This obviously, yeah.
Is it a tunicate?
It looks like a potato.
It does. It really does.
Potato.
I'll jot that down in the
Oh, there's a
log.
little shrimp friend in the
I don't
view
know
as
what
well.
that is. I haven't seen the movie still, so can't do it justice.
Which movie?
The Martian.
Oh, yeah.
All right. Well,
There's potatoes.
Lots
like
of potatoes.
most of the really good quotes in that movie
All
have
right,
swear words in them.
it doesn't
Is the
look like it's
unfortunate
living
It's,
anyway,
yeah.
so and it doesn't look like it's the right kind or shape of rock, so we're just gonna leave that there.
Cool.
Sometimes a potato is just a potato, but it's not. It's probably
It's
not
not
the
that
case
deep.
here. I'm not. It could just be a discolored rock.
Steve
you know,
wanted
maybe
to stick up here somewhere. I think it's dead. Yeah, it's an old piece here.
as much as I would like to go around and poke every single thing, it would eat up a lot of time.
Yeah, we gotta use our time wisely.
You still want to zoom on this dead one?
No, it's a sponge, sponge rubble. Move on.
Oh, there's a whip over here.
Mm-hmm. Yeah, we can snap zoom on that. I think it's one we collected on our past dive.
Roger.
Very cool.
You can snap zoom it from here.
Yep,
I'll just
you got it.
kind of do a flyby. Get that confirm
Oh,
ID.
it's actually
Oh,
it's a bamboo, but also one we've collected on a previous dive. So this is a bamboo coral.
Looks like some of the polyps are missing on it.
Yeah. This was one we collected on the very first dive. This is the one that had the very mucousy polyps. And we collected quite a bit of it, but it's very fragile skeleton.
All right, thanks, video.
Very fragile polyps. They
Yeah.
get stressed out and they mucus. Sometimes they bail out off the skeleton. But I think we did a decent job collecting it last time. We'll just note it for the record on this dive,
sounds good.
showing that it has a pretty broad depth distribution.
Yeah.
About 1,000 meters between. What was our first dive? It was around 2,700 meters.
Yeah.
Up now to 1,700 meters.
That's amazing.
Bamboo corals, of course. So the second family so far we've seen that are exclusively deep sea.
Oh, wow.
So they have lived and radiated or speciated in the deep sea. They didn't come from shallow waters. Well, they may have come from shallow waters, but unclear at this point. But a lot of the species we see on Earth today have evolved in the deep sea.
Very amazing.
Do they have any specific adaptations to that shallow water organisms don't?
Yes, actually. I haven't forgotten. I just want to take a look and see if there's any loose rocks here. So bamboo corals have relatively large polyp sizes relative to shallower water corals, because presumably polyps are very energy intensive things to maintain for an organism, but they're critical for feeding. So the thought is, in the deep sea, if you have more energy invested in your polyp, larger polyp size, you have potentially more efficient particle capture than if you had many, many little small polyps. And so they're mostly gelatinous, and they have some, but not a lot of defenses. But
sea star.
Is the mucus part of their defense mechanism, or is that like stress related?
I think it's stress related more, but they do have a tendency to mucus in the environment too.
Okay. Yeah, when you were talking, I was just like, so many lesson ideas were firing off in my head, like evolution, energetics, and like, oh my God, these are great examples to bring back to the classroom.
Yeah. Yeah, you'll find even fishes. So you'll notice.
Zoom in on this video, this little sea star.
You'll notice for fishes, a lot of animals have eel-like body forms because it's a very efficient way of moving through the water column and station keeping, because you're very streamlined.
Yeah.
You're optimized for energetic efficiency with the limited food supply you have. This is a Brisingid sea star.
Brisingid sea star, very cool. Yeah, all of these amazing organisms we're seeing, and just like hearing you talk about how they have all these unique adaptations and they've evolved to survive in these pretty extreme environments.
Got more
How
amazing.
do you, this one's pretty small and we don't
A little bit more.
have species level ID on these. How do you feel about suctioning this one?
Yeah, sure.
Woo!
All right. So normally these Brisingids are quite large, and so they're kind of unruly to collect, but this one is small enough that it might be able to fit in the suction hose. And so if we can do that, we can have a reference since they seem to be pretty abundant in their environment.
I'll float up just a little bit. You can zoom out.
Okay, come
And
wide.
then I'll float up. You can porch out.
Yeah, roger.
So this one's probably less than 10 centimeters arm tip to arm tip, based on the lasers.
Yeah,
and
definitely.
one of the things that I specialize in when I'm not here
Can I porch up?
is
Okay, yeah.
Oh, it's spicy.
the genetics and genomics of marine invertebrates in the
good.
deep sea,
Okay,
and specifically now to help support sequencing animals to build genetic reference libraries for eDNA. So you can think of eDNA as kind of a puzzle.
Yeah.
You know, the pieces and the way they fit together are the the sequences of DNA you might generate, but the picture of the puzzle are the animals. So
Okay,
you
yeah,
need both aspects to make it make sense in the big context.
I love that description, that kind of that analogy there.
I'm working on different analogies. Some people use the library analogy. So if you have a library and a bunch of shelves, the DNA sequences in your eDNA are, the books, but maybe the titles and the stories are your reference library, you know?
Right, right.
So there's a bunch of ways you can describe it. I like the puzzle analogy because
I do too.
we're doing a lot of puzzles here.
Yeah. Yeah, one of the ways we pass our time on the ship, especially during transits, is by doing lots of puzzles. I'm writing this down because I'm like thinking of a way maybe I could incorporate some sort of puzzle in my classroom, like a physical puzzle, and like illustrate
Looks like
this analogy about eDNA.
Yep. I've seen some pretty good lesson plans with Legos too. I can send you those.
Yes, that would be amazing. I'd love that.
It's cool to think about eDNA being taught in like younger classrooms because it's such a new technology. Like I feel like I didn't hear of it significantly until I got to college.
Yeah, I don't think I did either. Probably even after college. I'm just a tiny bit older than you.
Very similar time spans here in college.
Oh, I got one of those. Oh. It's not great.
I think if you can scrape up the center disc, the rest of the arms might come up.
Yeah, I'm trying to.
That's also a defense mechanism.
To grab on, to hold on.
He's like really kind of
taking him in pieces.
Get in there, come on, friend.
There it
Nice.
is. Okay.
Do you want the rest of his arms?
Yeah, that's fine. I think we're okay.
Okay, I killed suction.
ROV, which
Maybe they'll
jar
regrow.
is this going into?
Yeah.
This is jar 1.
Thank you very much.
So, can sea stars
Nice.
regrow from just like a single arm like that, even if the disc is gone?
Is that jar 1?
Yep.
Yeah.
That's so incredible.
I have trouble seeing those numbers. Alright.
So it just creates pretty much a genetically identical copy.
I did not realize he would
asexual
shed
reproduction
arms.
almost or something.
I know, it really popped off, didn't it?
Yeah, this is a process called we witnessed called autotomy.
Okay.
Can I try
And,
blue buttoning off? I
the
just want to see
ability
if it
to
sags again.
Sure.
I don't know
break
if anybody's checked on this
parts
ship,
of
but-
your anatomy as a defense against predators or disease.
Yeah.
You regrow them. We see them with regrown arms all the time.
Alright.
Super cool. Yeah,
I got some
as
tether.
I think I've told you, my background was actually in herpetology,
come down
amphibians
to
and
it. Came up,
reptiles, and
so-
so we see that a lot in those species as well.
Nice sample.
Yeah, just kind of going back to what you said, Leela, about how it's cool that younger students are able to learn about these, emerging new technologies like eDNA.
Is that on the second
When
one?
I was in high school, I don't even think we knew about eDNA yet. I'm not really sure. I'm not going to reveal how old I am. I know you want to know. But yeah, I think that's like one of the coolest things about science is we're always discovering new things and learning more and kind of modifying our previous theories when we gain more evidence.
Well, it's a new approach, and it was originally more widely used in freshwater and shallow water systems, and I think In the deep sea, it's been much less applied, but it's increasing for sure. It's now becoming, you know, a standard part of oceanographic expeditions for the deep sea, but that's new, right?
Do you know when people first started learning about eDNA or first started using that approach?
So there was, I think the approach was related to advances in DNA sequencing technology. Back when I was a graduate student, we used to just sequence a specific one specimen and get one sequence with what we call Sanger technology. And now we have high throughput technology where we can do bulk environmental samples and send in a sample and get, you know, 100,000 or more million sequences from a given sample. And, you know, microbiologists applied this approach first, right? And for them, eDNA is the actual organisms, right? For us animal people, we're looking at the fragments, right? And then it sort of grew from there. And, you know, my first attempt, or I wouldn't call it attempt, we were successful in 2018. I used to work, we had this Twilight Zone program where we focused on the mesopelagic. So my background comes with the water column, the deep pelagic water column. And one of the first thing that I noticed was that the DNA, eDNA was very dilute in the environment. I'm like, you know, we have to do better with the sampling. And so that's what's led us, that was the motivation for us to develop this autonomous sampler with large volume, you know, and we showed with a prototype that, you know, when you sample larger volumes, you find more things.
Find more things, yeah.
sort of an obvious finding, but, you know, we showed it. And, you know, and then with our sampler, you know, we can put it on a variety of different platforms. You know, again, we started with Mesobot. For those of you who have seen prior Nautilus cruises, we've deployed Mesobot on multiple Nautilus expeditions. And collected eDNA with our samplers for that, but now we've expanded.
Thank you.
Moving deeper, moving to the seafloor.
So amazing.
Yep.
It's a nothing. Sorry.
Is it really a dead sponge?
All right, it's a dead sponge with a crynoid.
Say one more time.
It's a dead sponge with a crynoid. Actually, there's a bunch of these hairy things coming off the top are actually arborescent foraminifera. Which are a type of single-celled protist animal, or single-celled protist, not animal. A bunch of brittle stars up there too, but...
Next video.
Yeah, all right.
Over the next one. Should we get another move in?
Happy, keep moving, yep.
230, co-pilot might turn to port just a little bit. Yeah, perfect.
Very cool. As Annette was saying just a second ago about this cool new kind of emerging eDNA technology, she was telling me one of the first days we were here, I think it was after you were talking, I was saying how I really want to bring this back to my classroom, and I also run science fair at our school, and I think it'd be so cool to do that. And there's actually a citizen science eDNA sampler, which I'm so excited you told me about, because I am totally going to try to get one of those into the classroom so my students can not just learn about science, but authentically do science.
There's a lot of different samplers out there, and we have a lot of different marine environments, and so there's no one size fits all sampler. So, you know, the ones that I use are you know, motivated by deep sea, you know, exploration and science. But the citizen science, it's a Smith root sampler. It's an amazing approach for when I did some work on the Fraser River in British Columbia, and it was it worked super well.
Yeah.
And in rivers, we're we collect, here we're collecting hundreds of liters of water to filter. And, you know, in a river, you know, we were lucky to filter a liter because of all the sediments.
Right, yeah,
Right?
definitely.
And so
Especially in like more
the filters
polluted
would
rivers
clog.
too.
What's that?
I said especially in like a more polluted river, it probably has a lot
I
more
suppose, yeah,
sedimentation.
I think that was not, you know, we
Not
you know,
saying that the river you
okay.
sampled was polluted, but just saying like, I know
Yeah.
that, you know, pollution is, you know, kind of a problem all around the world. And so
Right.
where I'm from, we see lots of sediment pollution. And so,
Okay.
yeah.
Okay.
We just had a question about how eDNA is different from DNA. And it seems like smaller fragments are based on the puzzle analogy, but what are the puzzle pieces made of? Annette, could you tell us a little bit about that?
So eDNA stands for environmental DNA. And so in the broadest sense, it means DNA in the environment, right? And that could mean anything from DNA in organisms to DNA from little particles, tissue fragments, and little pieces of things, fecal pellets, that sort of thing. You know, for microorganisms that are single-celled, that may mean the organism itself, but for animals, which, you know, is the focus of us on the biologists on this cruise, we are interested in, you know, the corals and fishes and other invertebrates and all that, you know, we're looking at the little traces of biological material they leave behind that contains their DNA, right? And so that's when we use the term eDNA, that's what we're thinking about, right?
Right, yes.
And so
it's kind of the DNA they left behind.
Yeah, so it's for that I think of it as a forensics, right? Where
Totally,
you know, you go to a crime scene, you know, people leave behind their, you know, cells, hair, whatever fragments of cells, you know, animals, you know, in the ocean do the same thing. So,
awesome.
Yep.
Yeah, just had a couple comments. I'm not sure, these might be my students making these comments or maybe fellow teachers, but they're saying, Mr. Matt Holden, you need to bring these back to Fayetteville for your students.
Oh.
And I will. Another interesting comment. People are wanting to know how often do we find species that are completely novel to science? I know, Steve, you've been on many of these dives. Could you tell us a little bit about that?
Yep. So typically, so it's a process to describe new species. But just for some of the cruises that I've been on recently in the past 6 to 10 years, we typically find at least one, if not more, new species per expedition.
Oh, wow.
Can we zoom in? I'm just curious what this is.
Often is a process that takes several years.
Okay.
To do.
Well, if you discover that we have found one, you have to let me know in the future.
Oh yeah, yeah. It's like some sponge rubble.
It'll definitely increase my coolness with my students if I was on an expedition that discovered a new species.
Yeah, it's a process. The field of describing new species is called taxonomy and systematics. How you name a species and how you differentiate it from others.
Okay.
And it has its own rules actually. There's a committee that has rules for how to name a species, and
there's a fish tray at the bottom of the screen.
Oh yeah.
Sorry, Steve. Shout out to Mariana for the spot.
Oh yeah.
Nice, good eye.
It's pretty little.
Is that the same fish we got from the last dive?
It's a cusk
it looks
eel,
like it. Yeah, it looks like. Yep.
Sorry.
Steve, it has that elongate shape you were mentioning is optimized for bioenergetics.
Yeah, it could very well be the same species. It has that dark, very dark pigmentation.
What kind of fish is that again?
It's a cusk eel.
Cusk eel. Very
in
cool.
the family Ophidiidae. You know, we did suction a fish very similar to that yesterday.
Yeah, I remember,
ago. Okay.
It's very cool.
Nice, yeah. Okay.
Thanks, video.
Normally we see cusk eels, they're much bigger. They're usually sometimes 30 centimeters or more.
Oh, wow.
They get very large.
So I do have to give a quick shout out to who is sending those questions in. It is my best friend that I met in college. She is a professor at a University of Arkansas. Thank you so much, Dr. Jennifer Davis. Love you so much.
Yeah. Copy. Sounds good. A lot of these stock sponges here, huh?
Yeah. And they're pretty much everywhere. I know these have been collected before in the North Pacific and Central Pacific. Sponges often don't get a lot of attention. The taxonomy is pretty difficult. There's not many experts in the world that can differentiate different species of sponges, especially glass sponges. But we have lots of collections, especially made by this ship, Nautilus, over the past several years, and others have been collecting glass sponges pretty widely. But like I was mentioning about new species description, it typically takes between 10 and 15 years, unless otherwise prioritized, to describe a new species from an expedition. And that's largely related to constraints on expertise and funding for this type of species biodiversity work.
Yeah, gotta have money to be doing this cool science. Support science funding, whoop whoop. I just got a question that came in that maybe one of our ROV pilots could help if you have a chance. Would now be a good time?
Sure.
So if you want to introduce yourself first and tell us who you are, and then the question was, how hard is it to become an engineer or someone like yourself working on Nautilus, or maybe you could give us a little bit about how you got into working with Nautilus.
Yeah, I can start. I'm flying Hercules right now, so the ROV pilot position. My name's Levi Unima, and I'm coming from Rhode Island. This is my first cruise flying this ROV. And I started in electrical engineering. Nothing ocean related or anything. Making car mirrors in Michigan, and then my high school science teacher from Washington State wanted me to get into this line of work.
That's awesome.
He was on the nonprofit for a exploration group, and then one thing led to another.
That's great.
Flying robots for like 8 years.
I love it. You never know where life's going to take you, you know. Didn't go into this field knowing you'd be here, and here you are. Rye, would you mind introducing yourself?
Yeah, I'm Rye. I am sitting, oh sorry, my fidget spinner just came apart. I'm sitting in the Atalanta seat, which is the kind of bird's eye view that you see. And that's it.
Cool. Thank you so much. Yeah, so as you heard from Levi and Rye, we have 2 ROVs in the water right now.
If you'd like.
ROV Atalanta and ROV Hercules, and they work together in a dual body system. So if you look at that bird's eye view, you can see that coming out of Atalanta is that tether that's connected to Hercules, and then Atalanta has another tether that goes up to our ship, the Nautilus, and they kind of work together. That allows Hercules to be decoupled from the ship so that it can move very precisely when we are taking observations and collecting samples, and that way it's not just like bouncing around like a tea bag would in like a glass or a cup.
Copy that.
All right, nice crynoid.
We can probably keep going video.
Yep.
Hear a lot of chatter on this one.
So one of the other characteristics at this site we noted when we first got down to the seafloor on our, I guess, last dive, but earlier on this track we're on, is the current was very strong in the area of waypoint 1, and it tended to see more sponges and corals down there,
Let's
and
do
it's
it.
dropped off a bit
Yeah, we could probably do another one.
as we've started to move upslope here. But we're also, if you zoom out and look at the larger topography of this site, we're actually kind of in a bit of a canyon. It's a very shallow canyon, so that could restrict water flow,
Some of the flow, yeah.
across the landscape, and which is why we're seeing more increased sedimentation in pockets
Gotcha.
that we weren't seeing a bit deeper.
And kind of settling out there in that
But
little
yeah.
mini canyon or something.
I suspect as we move up through waypoint 6, 7, and 8, it'll open up
that.
again.
Nice.
Because we'll be getting to the top of the seamount.
Yeah, so as
Pink
Steve was
thing?
saying,
Go for
maybe
the
the
pink
water flow or the currents are not moving as quickly, and many of these organisms
I
are
think it is.
Do you think it's
collecting
a cucumber?
their food sources
you tilt me
through
down?
that current as it kind of
Video,
passes
come
by
here, big
them,
ball.
and so oftentimes we find more organisms when we have a stronger current because they have more opportunities to collect their food.
That is, what is that? Oh,
Oh, it's just a,
Is it a cucumber again?
yeah, we're just looking at it on end.
But
It's
it's
like
just
rolled
like-
up like a ball?
yeah.
It's getting ready to take off.
Looking at its butt.
They're getting creative
is
about
that
getting
a-
looked at.
Sponge stalk
Alright, we'll
next
let
to
go.
it?
Yeah. Cool. Yes. Yeah, we've seen quite a bit of
Cucumber.
those
Just a cucumber.
Of the Q variety.
Sponge debris.
sponge
The
rubble,
cucumber
technical
accents
term.
the
Sponge
water
leftovers.
in such a way.
Mariana, we've got someone on here watching you. Wanted to say hello as well. I'm not sure who it
Well,
is.
hi.
They just said say hey.
Hello, hello.
That's so ominous. Watching you.
Someone's keeping an eye out.
Reminds me of the song from the Police.
Every breath you.
I'll be watching you. It's
Guys,
like
for the rest of
ultimate
the
creeper
stream,
song.
Rye and I are going to do a cappella covers of
I love it.
police songs.
I guess we could continue on with our last couple introductions. Gabby, would now be a good time for you to introduce yourself?
Absolutely. My name is Gabby Espino. I'm sitting in the nav seat. I'm also a seafloor mapper on board the vessel. I'm from Kailua, Hawaii, and just graduated, got my master's in oceanography from University of Rhode Island.
Awesome. Congratulations on that.
Thank you.
And Dan, would now be a good time for you to tell us a little bit about yourself.
Yeah, absolutely. My name is Dan Kirilov. I'm sitting in the video engineer position. I'm out of Seattle, Washington, and I regularly freelance for engineering for live production events, and I used to do a lot more builds for TV stations and that kind of thing, and very happy to have transitioned into this and happy to be here with everybody.
Yeah, we're so
Should we
happy
get
to have
20
you.
more meters in? Yeah.
Again, another, cool story about how you got here. Didn't go
Oh, no,
into
we're
this
still
field
moving.
or your field
We're
to
still moving.
be out
Got
here
it.
on EV Nautilus, but found your way
Sweet.
here. I love it.
We got it. Turn right just a little bit. You're watching Nautilus Live. Back to you, Matt.
Need a sign off.
Like, subscribe, and what do they say?
Don't forget to hit the subscribe button.
Oh, yeah.
Yeah.
Or your eyes on the bottom of the ocean.
Yeah, we totally all need to have sign-offs. I love it.
This is Nautilus Live.
We need that like breaking news sound, but then it's
Breaking
just a sea
news.
cucumber. It's like breaking news. Another sea cucumber.
What is that?
This is the
It's solitary
EV
hydroid.
Nautilus.
It's definitely a solitary
Where
hydroid.
you got it?
I'm.
See that, Steve?
We're gonna have to keep
What do
doing
you
that. I
see?
just love it too
Solitary
much.
hydroid, right here.
I still.
Earlier this year, we saw
Oh,
one that was
yeah,
like,
yeah.
there was like three.
Oh,
like,
yeah, there it
like
is.
4 feet
Yeah.
tall, or 6 feet.
All
It
right.
was massive.
Yeah,
This is a little baby one.
yeah.
I'm going
Yeah,
for the kill
I
here.
see what you're looking at.
Still trying to find it.
It's alright.
Oh, there it is. I see the shadow now.
Mm-hmm.
Oh, I'm coming in pretty hot. Watch out. Okay, zoom in. Yeah.
Ship moving complete.
Roger. Tilt down just a bit.
Roger.
Great one, Gabby.
Boom. Hi, Steve. What is this?
So this is a solitary single polyp hydrozoan.
Wow.
I wanted to look at us.
It's really
yeah,
pretty.
it'll come around.
Oh, perfect.
Whoa. It
Oh,
heard
it
you.
heard you. Oh,
Mhm.
It
It's a
did.
bit creepy.
Do we have
That's
speakers
satellite
coming out of
dish.
the Herc right
Oh,
now?
Herc is
Hello.
very loud. Herc is very noisy in the water.
Something in there.
Yeah, sometimes they have polychaete worms that live on and around the mouth parts. This one, I didn't see anything immediately. This one's pretty small. What do you feel like suctioning this up?
Roger.
I
You
feel like
wanna-
suction.
You want to zoom out a little bit? The ship stopped, right?
Ship is stopped.
It's small enough that we should be able to just noodle
All
it
right,
up.
I'm gonna come off the ground.
Noodle it up.
You can
Reporting
Yeah.
live
from
from over
So
1,700
this
meters
is
deep.
a single polyp hydrozoan.
Yes, so
Oh, sorry.
Are they attached to the ground pretty hard? We'll find out, I guess.
we're gonna find out.
I'm not convinced that suction's actually on and not.
Nope,
Alright, there you go.
nothing.
They're not often collected because they are big, and this one's not too big. So I'm hoping maybe we can collect it. The site's been pretty sparse, but hoping we can
You
collect
take
it.
the camera and do whatever you want. Yeah. I'm still
Yeah.
landing.
I can't remember ever
Are you
collecting
just
one
floating or
in
what are you
landed.
this
doing?
area.
Oh,
I'm
nice.
propped against the ground.
Awesome.
Finally.
Slipping.
Let's do it.
In the
Slipping.
well,
I might be slipping.
in the entire North Pacific, I've
Alright.
never been on an expedition that collected one.
It's super cute. It looked
So
at
we're
us and
gonna
then it
find
was
out.
like, oh, I'm shy. Gonna turn away.
I think base first, probably scrape it up. If you can do that.
Oh man, that jaw rotates annoying.
Got some people
Yeah,
in the
I-
comments super excited for this collection.
The
Take the knife. Don't take
the one
knife.
that
Alright, we're on Jerk 2, right?
that was
Yeah.
Yeah, it looks
mentioned,
like
there's
seen earlier
I see a
this
3
year,
on the other one.
generated a bit of buzz in my
Yeah.
office.
Just to confirm, I heard we're putting this in jar 2.
I think so, yeah.
Thank
That's
you.
a really, really
You want me to
large
start
one.
sucking?
Yeah,
That
that was such an
Can we do a
amazing
partial zoom?
image of it.
Absolutely.
I can
we were
do
getting
this.
earlier.
Yeah. All right. What would be the best method? Kind of scrape it at the bottom, and
Yeah.
'cause this is gonna
Let me know when you want it.
be a bit scraped.
Are you ready?
Yeah. let me
Okay, here we
just
go.
get- Yeah, get that
Here we go.
suction.
Alright, it's going. And I see some particles flying around in the bucket, indicating
Roger.
flow.
You might get bopped off the rock. if
Oh,
Nice.
something
That was a good one.
Okay,
got it,
and okay, I'm killing it. I'm killing
Alright.
it.
Good.
We're going to, it's
It largely intact.
largely intact.
Yeah.
Wait, there's a
You sound
still on the
really
ground.
cool as a
Yeah.
sea cucumber right now, Levi. You're cool and calm and collected.
That's fun.
And
Oh,
then,
there's actually
Great grab, Levi.
there's a sea star right here too, if you don't mind afterwards.
Steve, is this a Nidarian?
Yes. Yeah.
Alright,
So it could be in the genus Corymorpha,
One
which
of our
is
comments just asked that.
yeah. I mean it's common. It's a common genus, but honestly,
Here.
indexed
it's
camera
kind of a
back.
guess at this point. But it could be Corymorpha, but there are some small
over sea
minute
star.
differences.
Right.
What?
Oh, sea star.
Grab the
Right there.
Suction nozzle.
Oh, are you suctioning
Yeah,
nozzle?
I have indexed this to 3.
Oh.
Sea star? Oh, there it is.
It's a tiny little micro star.
I
Chris
still
Law.
don't see it.
I'll let you land first.
Roger. Okay.
Here, I'm just tilting my camera quickly.
You can move the main camera where you want, right? I'm over
I'm just making sure we stay in view. Alright.
Got it. I see what you're saying.
I'm gonna tilt down. Is that okay for you?
Yeah, perfect. Is that a good position for that little sea star?
Yeah.
So, good. Bam. Boom, bap, bing, bap, bam. Okay. All right.
Love that
I'm
song.
ready at 68%. We're on jar 3.
All right.
Hydrozoan.
Can we zoom first before the
hydrozoan.
Yeah.
suction?
I'm just gonna
Took
grab
the
this
words
section
right out of my
thing
mouth.
Okay,
Yeah.
ready?
Ready
Alright.
for zoom?
I'm just gonna go. I'm gonna do it.
This is a Goniasterid sea
Okay.
star.
I don't even see it.
I don't see it either.
Yeah.
All
I'm
the way to the right?
so confused.
Oh.
Oh, I was looking in the
It looks like a piece of candy a little bit.
Ooh, piece of candy.
All right, this is really small.
Nope.
Yeah.
Sorry,
It's
a little
adorable.
spicy on the zoom. There you go.
There we go.
This is probably a deposit feeder.
I'm guessing maybe when I try
They
to get
feed
as
on
much
sediments
of a zoom
and
as
small
possible.
animals in the sediments.
Okay.
You can go all the way in.
The
All the
before
way
this is
in.
the before picture.
It's also known as a cookie star.
Oh, look how little
Cookie
he is.
star.
He's
He's
got
moving
little
towards
feet.
us. He's moving
It
towards
looks
us.
like a little cookie.
Ever so slightly.
That's so cute.
Don't
Like
get
a
too
time
attached.
Is it like fuzzy on the bottom?
So,
Oh yeah, there's
Looks like
little
it's fuzzy on the corners.
appendages or something.
yeah, those are tube feet on the bottom.
Feet.
That's how they move around.
I want 2 feet.
All right, I think we're ready to collect.
Alright. You can come out video.
You can see it moving slowly.
So I
Yeah.
can see the arm and then
It's
So I will
cool.
say,
That's
in
good.
addition to corals, sea stars have been the second
Woo!
Quick and
most
easy.
commonly described group
Okay, I
of
killed it.
Alright.
organisms
Okay.
in this area.
No, this will
North
be
Pacific.
You
082.
see it make it into the
I
jar?
saw it. Yeah, it's right there. It's right
Okay.
there. Number
New
3.
species.
Jar 3.
Yep,
Yep.
there it is.
Nav, did you have another
Bam.
question, or was that?
Boom, bop, bing, bop,
Okay, great.
bam.
Thank you.
Good job, guys.
We got a waypoint. Oh yeah. She's
So this
playing
was
the
called a
birds
cookie
now.
star?
Yeah,
Porcelain.
it's in the, I'll put the family name so we can keep it.
Thank you very much.
Goniasteridae. And we might be able to put a genus to it up on deck. But a lot of the characters that differentiate the genera here are based on spines on their body and plates on their body and the arrangement.
Nice sampling, co-pilot.
You too.
Good job sampling. From the field, we have Rye and Levi.
And remember, kids, this is teamwork.
Mhm.
Yeah.
Makes the dream work.
This is ROV Hercules.
Do you wanna? I don't know if I can cycle the jars on mine.
No, I think it's only right here.
Huh.
Mike,
I don't know. Our sea
coming up.
stars, Roger.
just so you know.
Yeah.
What's the next direction you want to go, Nav? Roger? Looks like she's already lined up there.
Yeah.
Just had a comment come in
What the hell?
that I wanted to address real fast. A student that is 17 years old and learning about building small robots, which is so awesome, and interested in becoming an ROV engineer, and wants to know if that's a realistic like career goal. Not speaking for the ROV pilots right now, but as an educator, I
Yeah, we
say
could get
go
them
for
in.
it. You never know if you don't
2-4
try.
there.
You know, sometimes you get rejections, but don't give up. Follow your passion.
Yeah. I mean, this is an expanding industry, so like I would say it's definitely realistic.
Yeah, definitely.
And
also-
yeah, like definitely if you're getting experience already with building robotics and stuff like that, that's a great way to start.
Which is so cool also.
Yeah, that's really
Yeah.
cool.
But I also feel like even if it doesn't end up working out like long term.
Mm-hmm.
Some of the skills and connections you can gain by following your passions in that moment,
100%.
even if you're not trying to make it your whole life, can be really, really valuable. So I think if you like have things you're excited about and want to go out and do, it's always best to give it a try.
I 100% agree. You know, the more you put yourself out there, try new things, take risks, the more you're going to learn. And that's what it's all about.
So I'm stopping pumps 2 and 3 right now and starting 4 and 5.
Heard, thank you.
Can record that. Yep, thanks.
Another crynoid.
Nav, could we get a pin drop for samples? Please hold. 083 and 084.
Oh. This is an anemone.
Yeah, let's take a look at that, actually.
Yeah.
Oh, snap the anemone. I think we've seen a couple of these, but
I just love anemones. I'm going to get it right the first time one of these tries.
Oh, sorry.
They're so beautiful.
I can tilt you down.
Nice.
There you go. It's got a little spiky hairdo.
Yeah.
Yeah.
Super spiky. All right, video. Thanks.
Nice. All right.
It looks like the tips are lighter color. Is that just
Frosted
is that
tips.
something else?
Those are called frosted tips.
They're probably just less.
From the early 2000s.
So there are pigments in the body tissues of.
Real quick, sorry. Multipuffer, just confirming this is pumps 3 and 4. 3 and 4 starting? 4 and 5?
No, 4 and 5.
Okay,
yes.
4 and 5. Thank you.
Yep.
What kind of sponge is this? Or,
It looks like something's floating along with us there. Like a
Might be
little
on the lens.
circle, little sphere.
A little schmutz
Yeah,
on the lens.
more. So this is a polyopogon sponge. Glass sponge. All right, Emily,
video
snap
zoom
zoom.
in on a sponge.
It's been one of
What
the
is
more
he
common
up?
sponges we've seen over the past few days.
Yeah.
They're pretty typical from the deep sea, deeper than 2,000 meters.
I'll tell you up a lot.
Sometimes deeper than 1,000 meters.
Whoa.
Very
The
hot.
up is
Snappy.
Super cool.
cooking.
This genus is found globally, but I'd say most common, I've seen it in the tropical latitudes.
Yeah, it looks
But,
super attached to the substrate there. Almost
It-
like it has
it is.
little hairs or something coming off almost.
They're glass fibers.
Glass fibers.
Yeah, they
Cool.
attach there and they just pump water for the rest of their lives
Love it.
and filter out particulate stuff and consume that, grow, get bigger. All right, good.
Awesome.
All right, thanks.
Rye, our audience is loving that frosted tips comment you made about the anemone. I love it too. Again, another hairstyle I expect to see this semester in my classroom. Anemone frosted tips.
Feel like you could do a whole fashion show of sea
Totally.
organism inspired hairstyles.
Oh my gosh, we have a fashion club and they do a fashion show every year. I'm totally going to see if they'll do like sea inspired this year. So Steve, if there was one organism that you would really hope to find on this dive or even this expedition, what would it be and why?
So I have been looking for several years for, one second.
Yeah.
No, those are pretty round rocks, forget it.
Don't tell Val that.
I don't think they would be the right rocks.
Yeah.
It's really crusty. Yeah, I'm pretty impressed in how much crust there is, even as we're getting shallower. I'm looking for a white translucent octopus,
Ooh.
affectionately known as the Casper ghost octopus.
Oh yeah. I think we have a highlight video of one from a previous expedition, maybe?
Yeah, it's probably on a past expedition. It's probably a new family of octopuses that hasn't been described, and normally when you get to broader taxonomic groups and they're new, those are kind of really big deal.
Yeah, I would love to see one too. That'd be super cool.
Annette, could I get the water filtered for multipuffer pumps 2 and 3?
Yes. Pump 2, 286-023.
Yep, another sponge and coral.
286-023.
Yes.
Thank you.
Pump 3 is 148360.
148360.
Correct.
Thank you very much.
On the left of that sponge, like attached to it.
Yeah, there's a brittle star right there.
Okay.
Want me to hold down?
Yeah. We're landing down. I'm coming down. All right, let's go for chrysogorgia, and then we'll go up to the spongy.
Some of these comments just make me want to like go, oh. They're just very grateful to hear from us and just loving listening to us, and it's just so great to hear that.
Is there a spot lobster in there?
This is and there's one.
Yep. Like 2 little guys?
No, 2.
Oh.
2. Yeah, 2 Europtychus crabs and the bottlebrush coral. We did sample a bottlebrush coral chrysogorgia very much like this earlier in the expedition. This is different from the one we saw down a little bit deeper. All right, that's good zoom on there.
We could probably tilt up to the...
and what was in there again, Steve? What was in the bottlebrush coral?
Oh,
2
sorry.
squat lobsters.
2 squat lobsters.
Thank
Lost
you.
you.
This is another ferronematid polyopogon sponge with a brittle star on the outside.
Oh, great zoom. Would that brittle star be eating the sponge?
No, it's probably just hanging on there.
Okay.
I don't think they're spongivores, but could be eating something on the sponge.
Yeah.
There's a lot of small animals like hydrozoans that might attach to sponges and could be eating those. But there's also a lot of brittle stars that are planktivorous, so you'll often see their tentacles streaming off into the water and capture things that go by.
Cool.
All right. Looks good. Let's keep moving.
All right.
Onwards and upwards.
Should we do another move? All right. Copy 240.
Do you want to look up more?
Sure. Ooh, something.
So again, those of you just joining us, we are on dive number 3 of Expedition NA174 in the waters of the Republic of the Marshall Islands. We are currently exploring Wadajabato Seamount and looking for new species, range extensions, also looking for geology samples to tell us a little bit more about the history of the Earth.
This, we're just passing now this bamboo coral. We actually sampled this and its squat lobster host or associated animal a bit deeper, a couple dives ago. So this was the one that I was saying was excessively mucousy.
Yeah.
And you wouldn't really notice it here, but remember the temperature that these animals are living at is
oh wow.
nice
Oh my gosh.
macro.
That's
Oh,
amazing.
that's so
You got the
pretty.
macro
Those little
shot.
white balls in there.
Great.
Wow.
Yeah, those are probably reproductive cells, so those are eggs.
Oh, wow.
Yeah.
That's so cool.
Do they work their way out? Like some of the polyps seem
Yeah,
to have
yeah. When they spawn, they'll typically come out of the polyp. And they're expelled.
Wow.
That is so incredible.
Actually, it's kind of neat. On the upper one, there's
Yeah, you
some
could-
at the
that's
tentacle
what I
tip.
was talking
like
about.
kind of
Yes. Yeah.
Here, I'm gonna bump you up a little bit. Sorry.
Where's a node?
We need to turn
Oh,
that
there's
down.
another squat lobster.
Oh
there.
yeah,
There you
the
go.
little darker part,
See the
that's
darker
called
part?
the node.
There's a node, I think, right?
Yeah.
Correct
So
me if I'm
those
wrong.
eggs look like they're moving up
Oh, look
and
at look at
outwards.
those white balls in the left polyp.
Yeah.
See that?
That's why I asked.
Yeah,
I
from
didn't
the
realize I saw
tentacles.
the ones at the base, but I didn't see
And
the
then
one
what
on
are
the
those
tips.
pointy?
The ones at the
Oh.
base base of the polyp are probably immature, and they move upwards and outwards of the polyp when they're getting ready to spawn.
Okay. What are
Sorry,
those
Are they
pointy?
hermaphroditic?
There's a squat lobster there too. There's a few different squat lobsters. So there's a squat lobster genus called Heteroptychus that's most commonly associated with these
hello.
types of corals, and
Hello,
it's a
man.
different squat lobster than
They're
those
waving
bottle
at
brushes
us.
we saw
Okay.
earlier.
It's like see-through, just like the coral is almost. That's
Yeah.
so
Oh,
cool.
the little
It's waving
Very
at
nice.
squat
us.
lobster.
That's really cool.
Hello.
To see
Hi,
the,
friend.
It blends in so well.
Uh-huh.
Would that be on purpose, Steve?
yeah, there often are specialty appendages and morphologies to those appendages for certain species of associated animals. We term the likelihood of a species to choose one host or remain on one host. It's called fidelity. So whether they are going to be found on only one species of coral, or maybe they're found on any species of coral.
Okay.
So this particular genus seems to have a high fidelity to bamboo coral.
Very cool.
Yep.
And those pointy structures kind of coming out of the middle, what are those called, Steve? Like out of the coral polyps?
Yeah, so those are the sclerites. So if you see like spiny structures that come out of the polyp that stick up and down, those are sclerites. Those are calcium carbonate, usually needles or rods that give some rigidity and structure to the coral, and they provide diagnostic characters that we would use to describe the species.
All right,
Very
you can
cool.
zoom out.
Steve,
Right.
are these hermaphroditic or
No,
separate?
they're dioecious, so
Okay.
whole colonies will either be entirely male or entirely female.
Okay, so that's a
Yeah.
female, I guess.
So
You want to go for the big squats? You can come in.
Yeah.
Do they like expel their eggs seasonally, or like
That's
we
a
have a
good
question
question.
if
Yeah,
it's
that's-
related to the
You can
moon
zoom
at
in.
all?
We're going to go for the big squat
no,
lobster.
that's one of the questions that Finney is trying
Yeah,
to
let's
answer
go.
with some of the sampling that we've been doing.
Look at that.
Gotcha.
As an
So
adult.
this
Look at that one.
observation seems
Heteroptychus.
really important in terms of understanding the reproductive biology. You know,
Yeah.
I mean,
Yeah.
it seems really rare
Yeah.
to be really lucky to
We
catch.
often see,
Right, it's incredible.
right?
yeah, we often see reproductive cells
that is
in
cool.
corals, and it's
Okay.
not unusual to see the reproductive cells, but to actually see them spawning is something really undocumented, especially out here. But
Yeah.
it's been observed in stony, deep coral reefs in the North Atlantic before.
Where the eggs are actually on the tentacles like this.
Yeah, well, when they spawn, yeah,
Okay.
they release
Yeah.
eggs and sperm into the water column at the same time,
Okay.
typically. But the
Stretch it
seasonality
out.
or the periodicity
cool off
of
the,
it is
proportional
You should
control.
definitely pull that out.
well understood.
Okay.
You might have to go over your tabs.
And so one of the things that Finney is doing with the sampling that she's been doing is she'll be able to take those coral polyps back to the lab,
Oh,
and you can actually slice them very thin and look at them under the microscope
he's hot.
and look at
right?
the
So I gotta cool it off.
maturity of the gametocytes,
Yes,
so the cells that
right.
produce
Yeah,
gametes. Yeah.
Up is
Tell us a
still
little bit
three
more about the
chunks.
reproductive history.
Yeah, their size will vary depending on how close they are. That's a really nice view of that.
Yeah, it is.
I wonder if
great camera work
this
over there,
proportional
Dan.
control is when you click the button here.
Trying to catch it waving at us.
It is. It's happy to see us. Hello, friend. Oh, it looks like it even has little hairs on its claws.
Yeah, it's just not even going.
Yeah, a lot of crustaceans will have sensory hairs, if you will. But
look at that.
Yeah, they can sense vibrations, pressure
well,
changes.
let's remember that we're looking at this and we see it because of the lights of Hercules. That these animals are living in the dark.
Sorry, we're playing with our
so
cameras.
they need some way to sense their environment.
We can see that video. Sorry, we're getting sick back there. We're changing the settings.
So cool.
Very nice. Yeah, so you'll find that a lot of things in the deep ocean are red or pink because that wavelength of light is...
One of the buttons is chunks.
Attenuates very rapidly with distance in the water. So it's going to appear either invisible or black.
School buses.
Right.
Continuing.
But it appears red to us because we're bringing lights with us to the point of pigmentation.
Oh man, that was amazing. That'd be so cool to observe a spawning event. Have you ever had the chance to observe one?
I haven't. I have some suspicions that I may have, but
for sure.
We're still trying to confirm that. If a couple of years ago, or more than a couple of years ago, it's probably 2021. I want to say it's NA174-087. We observed a very high density precious coral community in southern Papahanaumokuakea Marine National Monument. And the water, so these precious corals are normally pink. They're the same one that we sampled a couple dives ago. And we were seeing this pink hue to the water column, and oftentimes egg cells, will have the pigment of their parent colony, and there was just this pink luster to the water around it. And it's tough to tell. They're very small microscopic cells, but it's a suspicion that maybe it was a spawning event. But
that's really cool.
We did take some eDNA samples there, so when those get analyzed, maybe we'll notice a very high proportion of hemicorallium reeds that might indicate that it's
give us some evidence that it might be spawning. Super, super interesting. We have a question about, I know that we are not in shallow water right now, but do shallow water corals tend to spawn related to the cycles of the moon? Do you know?
Yeah, I believe so.
Okay.
Yeah, they have more seasonal periodicity than stuff in the deep sea does.
Okay, that's what I thought too.
Yeah.
Cool.
The environment down here doesn't really experience temperature changes on a seasonal basis as the shallow water environment does.
Snap zoom this video.
There are other triggers
stock, pretty hard to see,
that they experience, but they may not be as sharp as in the shallower ocean.
Makes sense.
Come off it? What am I looking at?
It's another small bamboo coral.
Yeah, it's just another one.
Probably the same as the one we just saw.
It's hard to see with the sand.
I don't know how you keep spotting these things, Levi. It's incredible.
For indulging me.
I always like to look for shadows. You can usually see the shadow before you can see some organisms.
Yeah.
Because the lights, we have a lot of lights. And it'll cast a shadow.
To be determined to get better by the end of this expedition. Spotting these amazing organisms.
It's a cool formation.
Very neat.
Those are pillow basalts, pillow lavas.
Looks like maybe something down in that little hole. Almost, the little indentation there.
Yep.
Well, you want to snap the crynoid just to double check? Yeah.
Yep. Similar crynoid we've been
Yeah, we'll let
seeing.
it go. Thanks.
Crynoids also will break off their arms. They're very difficult to collect. Usually we try to collect them on attached to things already, so we can just pick up whatever they're on.
Gotcha, yeah.
If we suctioned it, it would just be arms and legs all over the place.
They don't want to leave home. Oh, that's a interesting, like, ridge almost.
Yeah. I expect as we get closer to the summit here, we'll have more of this ridgy parts.
Very cool. I hope it brings lots of organisms as well. Like we saw yesterday on those like overhangs and ridges, there were just tons of different life forms.
Yeah, that was interesting. I'm a little surprised that we saw more higher densities of animals deeper.
Okay, yeah, let's do another one. I'm stretched out, looks like.
Than shallower, but we're not quite at the summit yet, so there's still a possibility. We don't really have, I mean, I wouldn't, normally one of the things we do is we look at the marine snow and the direction that the marine snow is moving across the landscape to gauge the current.
I'm a little bit out of tether at the moment,
I mean,
out of leash.
Not seeing a whole lot of snow right here, yeah.
I'm gonna go for this one of the little spiral in it.
Ooh.
A little Dr. Seuss energy.
Yeah, totally. Looks like something out of the Lorax.
Yeah.
It's the same bamboo coral we've been seeing.
We going for that same up close?
Yeah, I'll get closer to it.
Okay.
Should have enough tether.
Yeah, we're definitely moving up slope. We're at 1663 meters right now. And again, we started this dive just a little over 1900 meters below the sea surface.
That's a tall one.
Oh, it's got the Scott lobster with his arms out.
Oh yeah, right in the top middle there.
I'm not sure which way he's looking. If he's looking at us or away from us, but we'll find out.
He wants his chance to be on camera too.
Yeah. All right, you can zoom in.
All right.
Let's see if this one has a similar reproductive.
Oh, there's two of them.
Yeah.
They fight.
Cool.
Are squat lobsters territorial?
Get out of here. Probably. I mean, you don't often see them on top of each other, but
He
you
only
have
has
them
one big arm though. See that? His right arm's gone.
Oh yeah.
I guess they are territorial.
There's definitely a.
No.
Just look at the other one and see if it's got
It
an
could have
extra.
been predation. It could have been.
Stole the arm. The other one's got 3.
Yeah, tilt up to the other one. You tilt me up, co-pilot.
Yeah, I can tilt up.
It should be right there. Yeah.
Oh.
See, he's got 2.
Yep.
Although they're not that big.
I'm just letting you know I'm coming up a bit because stuff in my sonar.
I don't notice.
I don't see any little eggs, but it's
a lot of reproductive cells here. That's one of the other things. I think the thing that I need to stress about the deep sea environment is that
is there like a meter?
life happens more slowly here than any of us could possibly imagine.
Right.
And synchronization in spawning may not happen all the time.
Oh, gotcha.
Okay.
Oh, so yours is 6 meters per division.
So
the right one, by the way.
Yours is 6
if you're not
meters per
close
division.
to your relatives, it may be
They swap
hard
sides from the last
to
dive.
synchronize spawning.
Right.
That makes more sense.
They're unaware, maybe.
Yeah, so they may just be
All right, we'll let this go.
independently
Independently
triggered.
Place here.
Gotcha.
You got it?
But ideally, for evolutionary reasons, you want to be in sync with.
Right. It would
the
promote
spawning.
diversity, right?
Yeah.
Yeah.
And one of the things that we find with these squat lobsters is it's not unusual to have several on a colony
and upward.
that may be very closely related,
Okay.
parent-child,
Uh-huh.
just to allow, ease and facility of reproduction.
Can we tell, like, if we collected squat lobsters, could we tell if they were, like, parent-child?
You could, yeah, there are molecular
Okay.
tools you can use to determine that. That's a field we call population genetics.
Yeah, very cool.
Or PopGen.
What is that?
Is that another anemone or
like,
something?
I'm gonna snap the white.
Yeah, let's snap that.
What is this? Is this of interest? Come in video. What is that? It's got a bunch of arms. What am I looking at? Dead spawn?
No, it's an anemone.
White anemone?
Yeah, anemone.
Yeah.
Oh, anemone.
Yeah.
Colorless anemone. Interesting. Alright.
Yep.
All right.
Another awesome haircut style. If you're looking for a new do, the anemone is super hot right now.
It's picking up a little bit here.
Interesting here.
Yeah, it looks like stuff's moving around, like through the water column a little bit more.
Yeah. So when we plan these dives, we're looking at grids of typically 40 or 50 meters. So
Looks like there's something on the right side of the screen there.
yeah,
Oh,
it's
yeah.
another golden coral, chrysogorgia.
Good. You want to snap on that?
The flabellate fan morphology, not the bottle rush. So parachrysogorgia.
Do you want a closer look, Steve, or not?
Quick snap.
Quick snap. You got it.
Everyone deserves their chance in the spotlight.
There you go.
Yeah, so
There's something in that one too,
parachrysogorgia
yeah.
also has a squat lobster in there.
Uh-huh.
Almost certainly, which is a different genus, Europtychus, from the
You tilt me up just a little bit.
other crab that we saw on the bamboo.
Thanks.
Yeah,
Oh,
nice. Okay,
little squat lobster. Hello.
yeah. This is parachrysogorgia.
Parachrysogorgia.
All right, coming out.
One of the cool things about Nautilus, if you're listening, is we can broadcast live all around the world thanks to our telepresence system. And just right now, we have people all around the world, which is super awesome. The Netherlands, Canada, the UK, Spain, Germany, Singapore, Ukraine, Philippines, the US, Malaysia, Japan, Australia, Norway, and Belize. So people all around the world are tuning in as we look for new discoveries in the deep sea. I love that.
Can you see where they're logging in from, or is this them telling you?
I can actually see where they're logging
There's
in
like a
from.
map.
So yeah, I can
Nice.
see
So
a map, and it shows
who's visiting the website, I think.
where all the current live viewers are around the globe.
Cool.
Can we collect this mushroom coral?
Good morning,
Yes.
world.
Right?
All right, let's do it.
I think suction might work on this one.
We're on.
Roger, you want to see your porch?
Yeah, you just porch it out for me.
All right,
Yeah.
I'll wait till you're
Yeah,
down.
bring in some porch out.
Great.
The other cool thing
Could
about
I
that.
get a could I get a quick zoom whenever we have a free
Yeah.
sec so I can get an in situ
Yeah.
photo?
Yeah, we will before we're
Cool.
just gonna land first.
No rush.
But
The
thank
other
you for the reminder.
There's also a little mini star. Wait, never mind.
there's a
That's
little
just sediment.
No, there's a sea star.
Is that
Right at the
yeah?
base
Right,
of the
right in front of the
door
porch.
there, see that? There's two actually.
Huh.
Brittle stars. All right, you want to zoom in on the?
Oh, yep.
Of course, as soon as I see that, my vehicle starts moving.
Ooh. What is that again, Steve?
So this is a type of mushroom coral. It's an octocoral.
Can you index the
All set on my end. Thank you.
can you index
All right.
this? Yeah. I couldn't remember what they were called.
There are two different genera, but honestly, I
I'll
can't
zoom
tell
out
the
so you can
difference
see
in
your arm.
situ, so we'll have to collect it to tell the difference. But there's the two genera that are common are Anthomastus and Pseudo-Anthomastus.
Okay.
Uh,
So like the false one, the fake one.
Steve
Yeah.
and not Steve.
Yeah, right.
the
Matt
differences
Damon
are,
and Steve.
Yeah, the differences are minute. Yeah. but usually morphological.
Yeah, we can zoom in partial on that. All right, I'm gonna start inhale. Here we go. Oh, here we go. Jar 4.
Yeah.
We're pulling in water.
So you're gonna
here.
see
I
the
can
polyps
Oh.
suck into the colony.
Oh, come on.
Get
You
in
might need
there.
to scrape it a little bit.
Peel the.
I have to
poor
kind
thing.
of scrape it off a bit.
It's staying pretty.
He
It's pretty on there.
does not want to let go.
Yeah,
Yeah, I'm going.
Get in there.
There we go.
Got it. Good job.
nice.
And 3.
So these
Can I
are
get in there?
these are taxa we know we can detect in environmental DNA
still
samples.
inhaling, by the
We
way.
found
Are
I
you?
haven't seen it.
ASVs or
Yeah, I'm still
implicon
inhaling.
sequence
I
variants
haven't
of
seen it come
mushroom
apart yet.
coral
Mm-hmm.
in
We had
EDNA in the
sediment though
past.
show up, didn't we?
It's definitely inhaling.
Using
Yeah.
a particular marker
Here,
that
I'll-
is specific to octocorals.
Okay.
You think did it go by and I just missed it?
Yeah,
Should I stop
totally.
inhaling?
Possibly, yeah.
You know
try
it's
that
working.
and see if you can
I'm gonna stop inhaling.
see
if
what it.
falls down.
So it's always good to have a reference
It's in the tube.
Yeah.
for
Yeah, I missed
Anthomastus in this region.
There it
There you
is.
go.
Oh,
Nice.
Just had a little moment in the wind tunnel.
It looks a little bit different now.
Well,
A little worse for weird.
Just
Wonder what could've
slightly
happened.
different.
so the polyps will reopen, right?
Yeah, maybe.
It's just sleeping. Right, Steve?
Yeah.
Yeah.
All right.
Do
Well,
we
no,
want
no,
that brittle
for-
star?
really,
That's right in front of the porch?
They're really resilient. The polyps will reopen.
You want the brittle start? You can come
over
in video, I'll
time.
get you lined up with the brittle.
All right.
Come in.
I
Right by
think
the rope there, of course. There's a small brittle star.
those you know corals, especially octocorals like that, we always talk about their feeding, but they actually have a lot of other functions that occur off there.
It's like, I'm getting out of here.
Yeah.
He saw his friend.
It does not want to be.
Actually, the brittle
Vacuumed
stars are notorious for jumping ship. As soon as you start taking a sample, they'll like throw themselves off of like anything,
Yeah,
anything you start
sometimes.
taking. They
Get
just
away
start
from me.
Jumping.
Respect.
I mean, this one just witnessed an adoption.
It's like an alien.
Yeah, it's
like,
going to tell its friends, guys,
away
guess
right
what
now.
I just saw? No one's going to believe it.
got beamed up into a UFO.
A giant bright light came and took my friend.
That looks good. We can move on.
All right.
I'm gonna get you squared up again.
One of the other cool things about the map that shows where everyone is viewing right now is it shows if they are returning
You wanna look up more than that?
viewers or new viewers.
Is it?
And
yeah, you
we
can
have
open it,
over 100 new viewers right now, so
Hey.
super cool.
Wow. Hello, everyone.
hello,
Welcome.
hello.
Hi.
Great. Perfect. You want to switch on 30? Ooh, what's that?
Switch on what?
Oh, there
wanna
might be
fly
a seep
in 30
down there.
minutes? Yeah, you want to zoom in on the sand sediment?
Yep.
Stock program? What do we got here?
Oh, I see what you're saying.
Yeah.
Yes.
Great.
Oh, it's a stalk, but with hydroids on it.
You want a closer look, Steve, or keep going?
No, that's all right.
That's all right?
Yeah.
Roger. So we got some tether. Yeah, I think so. Ooh, I see something up here.
What could it be?
What is it? Is it a bolsoma with a smaller head on it? It's pretty tall. Going for the kill.
We're coming to find you. Oh, there it is. I see it in that
This is
view
what I'm
over
looking
there.
at, this guy.
Yeah, I think this
It
one.
looks like a mushrooms. It's so cute.
I think this one might be Caulophacus.
Caulophacus.
Polyphacus.
I think we should do a.
Megan taught me how to
like a
spot this
trivia.
one. And she was like, now whenever you see it, you can yell Calophacus really loud.
Trivia competition.
Oh, I would fail.
Steve and Megan.
Can we go by like mouthfeel?
They
I'll
have
win
to,
on that one.
No, not
Calophacus, is that what you said, Leela?
It should be like a good
Yes. Don't ask me to
trivia,
spell it,
like
though. I will not know.
I will try my best.
We'll give
Alright,
you
let's get
incorrect
close and get the
information.
camera to this guy, video. Let's get it way up in here.
CAULOPHACUS. Polyphacus.
All right, I'll be
Yep,
ready.
I totally
Used
would have
it-
spelled it wrong.
Used in a sentence. Polyphacus is a genus of sponge.
Nice.
All right, good
And
to
can
go.
you give us
Pretty good
the
use.
origin,
Oh.
please?
Is there a different
Hand me down a little.
sentence you could give us?
Tilt it down, we'll zoom in, see what we got. It's coming right at me.
I used to love watching those spelling bees. I'm not a very good speller, so it was always fun to watch and
I'm slipping.
see how those
I,
kids are so good at spelling these incredible,
I wasn't as
difficult
good
words.
in spelling bees. I did enter the geography bee in my town.
Oh, cool.
Ooh,
How'd you do?
I went to the States, actually.
Oh.
Nice.
What is the definition of a peninsula? Or is it more like what countries where?
Yeah, it's more about like, you know, deserts and coastlines and
Well, don't they have the thing where it's
oceans.
Google image now? And it's a game.
GeoGuessr. GeoGuessr.
Oh,
Oh,
people
yeah,
that can find the
GeoGuessr is so
streets
right now.
fun.
It's like
So
they
you're
just
middle of
drop
nowhere.
you on a random Google Images, Google Maps
It's
road.
Street
and
View.
You gotta figure
Drops
out where you
somewhere
are.
on Street
Yeah.
View. And you get some.
I know a
you can,
lot
like,
of teachers that play it like daily
look
with their
around.
students. A lot of history teachers.
I love that game.
Very nice. Well, can we look at the backside and I'll show you some other characteristics about Polyphacus. It has a very unique attachment point.
I'll
You
bring
can
out a
kind of
little
see
bit
it in
first.
head.
Atlanta a little
wide
bit.
camera.
Yeah.
See the wide
Yeah,
camera?
you can see on the back side it has an attachment point on the back and
Oh, yeah.
it flares out concave.
I
Like,
can't
it's
really put the
concave in the
ROV
back
over there,
and
but-
yeah.
You think you're out of tether?
Well, it's because it's like downhill. Like
Oh,
that
yeah, you're
might be
gonna
like
struggle
that
though.
might be the best angle on it, right
So
there.
like from
Yeah,
the upper cam.
yeah. Is that a good enough angle, Steve, or do you
That's
want
all right. Yeah,
Okay. Sorry.
we'll get some. Yeah, we'll see more.
All right. We'll try to find that. Does that mean the
Maybe.
current comes from uphill, Steve? Like, does that tell you which
Uh,
way the current is, or does not necessarily?
I think I would judge it based on which way
It's leaning.
it's leaning.
It's
Yeah.
leaning down.
Most of the time when you get to the tops of seamounts, the currents are always downslope.
Yeah.
There's a little bit less sediment buildup here than even just immediately below us when we came on watch, so it may be a little bit more.
I see another sea
predictable
cucumber.
Yep, see another sea cucumber.
the color definitely stands out.
Yeah.
Want a little snap on that?
Sure. You can snap cucumber. Do spiky back. A spiked back cucumber.
What's that Pokemon that looks like a pink piece of chewed gum?
Oh my gosh. I can
Slowpoke?
see it in my head.
Which one?
Slowpoke?
No, I thought Slowpoke is the little guy.
Jigglypuff?
No.
No, there's another one.
Ditto?
Yes.
Yeah, Ditto.
That's
There
it.
we go.
Oh, yeah. I think I had that card.
This kind of looks like a giant tongue.
Yeah.
Yeah,
Yeah, right
like,
now.
Just saying.
Yeah.
Got a couple questions coming in about what is
Good to
life
bring
like
it up.
Nice.
on the ship and what a typical day is like.
Thanks, dude.
Yeah.
You should have followed along on my takeover yesterday on social media, but that's okay. We'll have plenty of other opportunities. But a typical day, most people on board have basically 2 4-hour shifts where we're in the control van, which is where we are right now, as we're watching
Ship is
live
stopped.
dives. And when we're not in the control van, we're either sleeping or eating or watching live dives in our social areas. And then we also do lots of crafts to pass the time and lots of
Another
puzzles.
crynoid there.
Yeah.
Yep.
I'm going for something. I'm going to keep going.
Can work out as well.
There's a sea star or something. We just passed it. It looked just like that one was
Crynoid.
earlier. Yeah.
Seen one crynoid, seen them all. No, there's a lot of different species of crynoids.
Oh, I see what you're aiming for.
Another sponge.
Can we
What's all that
look
stuff
at,
down there?
Can we look at that? Yeah, there's a bunch of rubble there.
It's like a
Possible rock.
No,
Oh, I see what you mean. The
Yeah.
like different color.
Yeah,
I like
different texture
looking at
almost,
rubble,
too.
rubble accumulation because it can tell you what's upslope sometimes. So these pockets and divots in the rock can give you some idea about what's going on upslope.
Hey, Steve, we just had a question. Do these organisms
Is there
Yep.
on the rubble?
have
Absolutely.
abilities to sense sound or pressure
Oh,
variation?
there's something swimming around in there.
Pressure probably, yeah.
Yeah, okay. That's what I thought.
Got a polyp
Oh.
there.
Yeah.
Okay, so any tighter?
Tighter? Absolutely.
It looks like there's something over on the left.
All right, I've seen this before, and you're not going to believe what it is. Go ahead and guess.
skeletons.
This dead
Muscles.
sponge.
Yeah, it looks like broken pieces of like shell to me.
You're on the right track. All right, we can zoom out and move on.
What
But
is it?
What? Yeah.
They're actually barnacle plates.
Ah.
I've seen this before around Johnston Atoll. We didn't know what they were. We called them crusty chips. They were
Yum,
iron
yum.
Are they
manganese
coming,
coated.
are they the barnacles growing on the rock, or are they coming from,
I think
like,
they come from
up
elsewhere
above?
upslope.
Oh.
But sometimes we wouldn't see them for hundreds of meters, but they would just accumulate in these pockets.
Oh.
But they're the plates of a barnacle are actually, I think they're called scutes.
Oh, like a turtle.
Yeah.
It should be called Bolosoma Mountain.
Bolsoma Mountain.
How about another 30 meters in this direction? And then we could start transiting towards waypoint 8.
Sounds great.
Yeah.
Sounds good to me. That one's very round. Like a perfect ball.
Yeah, it's like almost, like a perfect sphere.
It's like a dandelion.
No.
Yeah.
I am not seeing many rocks here. I'm
Yeah, it's almost just like a
so it's-
complete,
total crusted over.
yeah.
So the ridge, at waypoint 7, the ridge is going to turn towards the west.
You want me to snap that while we float by?
There you go. You can see the angle of this one.
Yeah.
Good to zoom as we pass?
Yeah, that's another Colophacus.
Yeah, it has that same kind of morphology as the one we just saw. I like the view of that slope. Very cool. Now we just need a really cool Casper octopus to come over that ridge.
We're a bit shallow.
Ah.
We typically find them closer to 3,000 meters and deeper.
Gotcha. Okay, well, how about a whale? Just putting it in the universe.
I was on the cruise that saw the sperm whale in the Gulf.
What? Were you in the van for that?
Okay. I was not, I had just gotten off watch actually. And so while that was going on, there were whales that were breaching on the surface in the distance. So I was watching that because I thought that was cool.
Were they
Yeah.
a different kind of whale, or were they?
They were sperm whales.
Oh, huh.
Incredible.
How deep was the ROV?
I think it was about 700 or 800 meters.
Okay.
So we were
So
doing
just like
water
cucumber.
column transects. During that time, we were
Wanna
not on the
snap on that?
seafloor.
Sure, you can snap cucumber.
And then
Sea cucumber?
afterwards, we can move back to the port a bit, to that rock pile, just to see if there's anything interesting.
Absolutely.
Cucumber.
Ditto.
Again. Not as vibrant as the last one. But still very tongue-like.
Yeah, when we saw the whale, I definitely saw some adults
Yeah.
lose their minds.
I'll bring it back
Oh, I
up.
bet, yeah.
Thanks, Vinny.
But it was...
Heard people
it
were
was a-
jumping
it
up and
was a
down
good-
on the couches in the lounge.
that's accurate. Yeah.
I mean, I don't blame him. I'd be doing the same thing.
I call that the Tom Cruise.
Yeah. This still looks pretty crusty in here.
Is that just a
Yeah,
pile of
sponge
dead
rubble.
sponges?
Yeah. So good. Just another cucumber.
Yep.
Oh, what's that? Little sea stars? Or one sea star.
That's it.
One
Chunky
Brisingid
cucumber.
star. I think it's the same one we suctioned.
Earlier, that
How
we
do you-
Same species.
got a few legs off
How do you tell that
first.
from a crynoid from far away?
Just
Experience.
It's an experience thing. Yeah.
He's closer to
I
the
see.
monitor than we are.
He's just that good.
Really, you
Okay.
can't confirm until you get up close, but
You come in, Vinny.
Brisingids
Alright.
have a symmetry and a definition to them.
Right. Like a crynoid wouldn't be as
Crinoids are more feathery
Nicely.
and
Look
fuzzy.
how the legs
Yeah.
change color.
spaced out.
That's interesting.
Yeah, they seem to be
Or
have a
maybe
different
they're just-
pigment closer
Is
to
there
the.
like something on
the
top of
disc.
the legs? What is that? What's
It's
going
like
on?
knuckles.
Yeah.
They could be regrowing too. Usually they have different color when they regrow.
There's not parasites on there, is there?
There are some parasites that live on sea stars.
That's on
yeah.
every leg, though.
She's got that like orange section.
Yeah, if you see they have, sometimes there's small crustaceans that can parasitize, so it'll have a little a growth gall or something like that. Yeah, it's not unusual. Small gastropods too, snails, can parasitize and cause wounds that might heal irregularly.
It's got too many holes in it. I don't like it.
Giving you the ick.
Yeah, he's got those little like. Yeah.
I think
Alright,
there's
where
like a
this will go.
word
All right, I can bring that back out for you.
too many holes.
Yeah,
Pores.
It looks soft.
No, looks too many holes in it.
I want to pet its fur.
You can come touch it later in the lab. They're actually very spiny.
Oh, not
It's
very.
it's not fuzzy. It's a
Oh.
spiny.
Not something you want to touch.
Not ideal. Yeah.
Does it have stinger cells?
They do not.
Okay.
No.
Is that sponge there?
Actually, can we look at this sponge? This one might be.
Yeah, that looks interesting. Like a little white dot right in the middle.
Yeah.
Is it an associate?
Nice
Oh, it sounds
terminology.
like-
Whoever said associate.
Plate.
It was me. I like
Nice.
associates.
Or is it like a vase?
Yeah, this is Atlantisella. It's a type of glass sponge.
Looks like a flower.
Mm-hmm.
Could I get a hint on the spelling?
Oh yeah.
Sorry,
Come in,
Steve.
video.
All right.
Outside land. Yeah, I'm on the ground now.
All right, you got it.
You tilt me down just a little
Yeah.
bit.
Oh,
you.
that's so cool.
It's in sea log.
How are those little holes doing for you, Levi?
No, this one's okay.
Okay.
Wonder what it's shaped
Can we
like.
suction up a little piece of this?
Yes.
It's probably going to be soft and like cotton candy-ish.
You said it's a glass sponge
Oh.
again?
Yeah.
I'll bring it out so you can see your porch.
So if we get a little piece of this in the suction hose, that'll at least help us get some tissue off of it. And we haven't
Off the
collected any sponges so far.
Are you off the ground?
Yeah, I'm
No,
off the
you're-
ground. You can porch out if you want. As much as you need.
Let's do that.
And then,
I'm gonna tilt you down so you can see. Here's your porch.
Ah, I just figured out what the fear of holes is, Leela. It's called trypophobia.
Really?
Yeah.
All right.
Well, that's what someone put in the comments, so we
there's
have
our
to
there's our
double check.
word of the day.
Word of the day,
that's
All right.
trypophobia.
So, bucket. I think bucket, we put something in 5 already, didn't we?
No.
Double
four
check.
I thought the little,
5 is empty.
oh, 5 is
Four.
empty. Alright.
Yeah. Partially vacant.
Perfect.
Should stop. So looking good.
Yeah.
Got a few more
give me a
audience
nice
members
section.
from new locations. Turkey and India have now joined us as well.
Is there in India? I mean, there's a lot of people in India, but
Yes.
that could be my grandmother.
Oh my gosh!
If it is, hi Ava.
If it's Leela's grandmother, please send a comment.
Oh.
I don't know.
It's like when I meet other people who are like, know someone in Canada and they're like, do you know Joe? I'm like, no.
Yep,
There's
I'll bring that
one
in a
million
little bit for you.
people in
let's
India.
zoom a little bit.
Right, yeah.
So we're only trying to section a bit of this, right?
Yeah, let's say, you know, a quarter of it. Let's see what we
Alright.
get,
Okay.
how it breaks
I'm all
apart.
the way right on my,
Yeah, that's fine. I got a good view.
Camera.
You got enough?
Alright,
Alright.
you want to bring section up?
it's on.
It's on.
Oh,
Okay,
yeah.
It's going
there
to
we
be
go.
pretty
It's
soft
sort
and cottony.
of
It is soft
Oh,
and
yeah.
cottony.
Yeah,
How much
Yeah, it
do you
almost looks like
Let's,
cotton candy.
about half of it maybe.
I'm trying to get little more bigger chunks here.
I think if you get
Scoop it
that
up.
much, that'll be good.
Yeah, okay.
That's about good. Yeah.
There?
Okay, I'll
Happy.
wait for that big chunk to get
Yep.
in the jar.
There you go.
Nice.
That's more than
it's
enough.
in the jar. I'm stopping.
Alright, coming back out.
Roger. So if I bit into this, how would I feel?
Sad is my guess.
Like,
Probably
would it be like eating
confused.
fiberglass
Confused.
or like,
Can we poke this rock?
yeah,
Poke it.
I'm curious. It's big enough that
we
it
may.
may not
I
be.
can move.
Oh, sorry.
Nasty,
No,
but-
I can get up there. I think.
Maybe if you can poke it, we won't have. Well, we can decide now if it's something we want to set up
Turn
for.
left. Like
Yeah.
that.
Sponges
Ask
are
with left,
peripheral.
And then get up and out of the rock.
Yeah,
Thank you.
there we go.
Yeah, this one's.
Bonk. Sorry, there's sound effects with this arm.
I'm
New
loving,
arm.
I love the sound effects.
This one?
Yeah. Does it move?
Goink.
It's moving the ROV, so what was that?
No,
No.
Okay.
that's
No.
the mountain.
Yeah, never mind.
Good idea.
Nice try.
While we're here. Alright, I want to float up.
I really liked.
Yeah.
I can't remember which ROV pilot it was, but one of them used to have a sound effect for the lasers that sounded like a lightsaber. You know.
That is a really good
That
idea.
is a good idea.
All right, Levi.
We can bring that back.
Is a good
Right.
idea.
Don't give us ideas.
Engage the laser scalers.
Love it.
Warp speed. All right.
So,
Oh.
could that sponge, is it
Oh, little shrimp buddy.
potentially going to die because of that, or would it
No,
recover?
it could live on.
Okay. Cool. Oh, look at that
Yeah.
shrimp.
Which bearing is that?
I'm just giving you.
240 again, or? Right. Yeah, that sounds good. Yeah, that sounds
I
awesome.
love those
All right.
shrimp. when
2-4,
they swim.
sorry, where we
So
go?
cool.
240. Should be your
Oh.
head still.
I'm already there.
No. Got plenty of room to go. I'm stretched out, so. Nice sample.
Mm-hmm.
Nice.
Again, for those of you just joining us, we are
Wanna remove jars
in the
and
waters
shove it over
of the Republic of the
vapor
Marshall
dryer.
Islands near Bikini Atoll, exploring Wadajabato Seamount. I have to look at the word and like say it in my head every time before I say it.
Question if I inhaled long enough on the last one. Can you put the jar back up?
Yep.
I guess I did see one chunk go by.
I saw a big
I saw a chunk
There
go
should be
by.
two
Go,
chunks in
go
there,
to
though.
the
Yeah, but
last
there
one.
should be two chunks in there. There's only one
Do you
in there.
wanna
It might be
turn
stuck
it
up
back
in
on?
the.
I kind of want to turn it back on.
Do it.
It might be stuck up in the filter. They sometimes get
That's
stuck
true.
in there.
I'm just gonna give it
Just like
a
for a
little
second.
bit. Just like a second.
And someone from Australia just asked what our robots are called,
Is
and
it
we
king or
have
jar just too far?
ROV Hercules on the pretty close to the
Alright,
seafloor, and
that's
then
fine.
attached just above it is ROV Atalanta.
It's probably stuck to the filter.
Levi, you know anyone in Davis, California?
Yeah.
They say hello.
Is that my aunt?
I'm not
Aunt
sure.
Jill?
If it's Aunt Jill, put it in the comments.
Friends and
I
Family
don't know if
Day.
I told her I was on
Identify
this, though.
yourselves.
Yes. Who are you?
What is your business?
Also, Steve, someone said that might have been sound effect Trevor.
Yeah, maybe. Yeah.
That does sound like Trevor.
Seems like he's got a reputation for this.
Yeah.
Trevor is the one who started Bonk, so like,
Okay.
yeah.
Can we poke that rock?
Yes.
Does look like a nice
That'll
one,
be
doesn't it?
$5.
And yeah, we are live 24/7, even when we're not doing dives.
There's that big
Can
round
watch
one
the
up
ship
there.
as it
Let's-
cruises
alright,
along.
alright.
Yep.
Let's go with per Bonk. Seems more lucrative for me.
Oh yeah.
I can bring it over a little bit.
Alright, I can look at the porch. Which rock?
He circled it.
It's this one here.
Yes. Right. Alright, I can just bring the arm out. I don't know.
All right, cool.
just find some
I'll
word.
collect that $5, Steve, and I'll get it to Rye shortly later.
Yeah.
I like how you're the intermediary for
Yeah,
this.
yeah. I gotta
Wait,
take
Matt,
my
are you
cut.
taking a cut? No.
He's the manager.
I don't need a manager. I didn't see that.
What about the one lower
Attached.
left?
Yeah, what
Yeah.
about the one to the left? It's
This one?
This one here?
This one?
Yeah.
It looks pretty round, but maybe I just want to try it anyway.
That's a
That's loose.
schmoomin' one.
Ooh, movement.
All right, why don't we pick it up?
Alright,
And
I'm gonna try to like
at least we'll get something else.
get it a little bit.
Oh, there's
out
that,
of here.
the bigger one's moving too. See
Oh,
that?
yeah.
You see that?
That's a pretty big one.
Let me
though.
move the ROV. Hold on.
Yeah, I'm picking the arm up.
I like the bigger one better.
That's a pretty
Alright.
big one though. You think that one's gonna fit?
Yeah. No problem.
Okay.
Oh.
No. Oh.
I'm also saying on
All right, go for
behalf
the
of
smaller one.
Wet Lab.
On behalf of Wet Labs.
I mean, I don't speak for Val, but that's a
Yeah.
lot of rock.
Alright.
Good little smaller one.
What, you want a little one?
I can't promise this is a good rock, but it is
Yeah.
a rock.
We'll
It's
just
a rock.
pick it up and we'll look at it. It's possible that
Nice.
it's just manganese, in which case,
Yeah.
copy. That's probably too small, isn't it, Steve? It's just a little manganese. Or is that gonna have rock on the end? We need to get like a rock saw down here and just.
Looks like something's on it.
Yeah,
You
can we
wanna zoom on
rotate,
it?
rotate
Yeah.
and zoom?
Look at my rock.
It's pretty round.
I'm getting indecision energy right now.
Yeah.
We want to try the
So
bigger
why don't we just stash it? Yeah.
All right, we'll stash it.
We'll stash it in the starboard box.
Alright.
Coming out.
Starboard
Just in
Yeah.
case.
box. Can I open it? Starboard
Yeah.
box open.
I'm
Or
just
do you
gonna
want me
get,
to- do you want me to wait till you bring her around? Whatever.
yeah.
You never know. Maybe there's something in there, but
I have so many pictures
All
of
right,
this
so then I
rock.
gotta switch cameras around a little bit for you. Right?
Sure.
I gotta get the
Yeah.
camera starboard bio instead of port rail. There you
Yeah,
go.
there we go.
All right. So now you're kind of in the blind
And yeah,
spot.
Levi, let's see, Jill and Bob,
Oh, hello.
they're watching from while they're having breakfast.
Awesome. Thanks for joining us.
As Selena in Seattle is also watching,
There
and
you go.
great job on the zooming.
Where would we like this rock?
one of the big.
Hi, Selena.
One of the big ones?
All of them are open, so
Oh, sorry.
dealer's choice.
Let's go E.
Okay.
Nice and E for easy.
That one is E.
That is my girlfriend shouting out, so.
Awesome.
Oh my god.
Yay.
Shout out. Was that Dan?
Shout out,
Alright,
yeah.
You want to
Shout
try
out to
closing that?
Selena
Make sure
from
it's
Seattle.
Copy.
closing. If I gave it
Starboard
a...
box close.
We've also got some Canadians watching, right? They say
Oh.
they're not family, but they're going to root for you like they are.
Yay!
All right.
Sorry, I'm just coming up on Atalanta because I'm a little bit close to you. Alright, I'm coming back around. Let's go.
I can bring the camera to catch
Steve, for the next ship movement and us movement,
Yes.
do you want us to go towards waypoint 8 now?
Yeah, I just want to go straight perpendicular to the contours upslope.
Okay,
Yep.
sounds good.
Yes. Alright. Put you to bed. There you go. Okay.
And then Nav just confirming that was 87.
Nice rock.
Data, you are correct. That's 87.
Lovely, thank you.
This is interesting. Is there like
Yep.
dust in the water column? See this?
What do you? Oh, sorry.
Look at this. Is this for me?
Just the sediment?
Yes.
Hello?
All right.
Yeah, it's just
Turn my camera
kind of
straight
stagnant there.
now.
Not much current.
Yeah, great. Good time. Mm-hmm.
Leela, your mother says hello.
Looks like
Hi, Mom.
maybe a little bit starboard.
I
of,
love it. Family day.
Maybe
Family
250,
day.
maybe? You want to turn
She is
250
watching
a
from
little bit?
Chicago,
Come to
Illinois.
starboard, just a little bit while
Yeah.
you're sonar up with it. Okay. Okay. Pretty close. I gotta check it here.
So I'm going to turn off 4 and 5 now and turn on 6 and 7. Leela, can you get that?
Yes, on it.
Okay. All right. 4
Could we
and
get a
5 off.
pin dropped nav for
6
samples
and
88
7
and
on.
89? Thank you, Nav.
You're watching Nautilus Live. We are 1,626 meters below the surface of the ocean.
That does have a ring to it.
It does. I love it.
A sea mountain where no one has been before. I believe this was freshly mapped too, right? Yeah, so
I
this
think
was
so, yeah.
freshly mapped by the ship, and shortly after,
First
here
ones
we
to
are.
put eyes on it. So cool about this expedition, like getting to explore places no one's ever seen before.
Very Star Trek.
Right? Our mission.
Oh, what is this? Right, this is the
Oh, yeah.
gelatinous on the
Yeah.
rock, lower center.
It's a megalodocopia.
Right,
It's a
down.
tunicate.
You come in, I'm on the ground here. What is
Multipuffer
this?
confirming this is going to be pumps 6 and 7.
Yes.
Thank
6
you.
and 7.
What kind of tunica did
Is
you
there
say that
something
was?
in
Oh.
there?
Sorry, those just ended. Yes.
Was this a tunicate?
This is a
4
tunicate.
and 5
It's a
ended. I
carnivorous
ended
tunicate.
them. And so 4 says off, but it appears to still be running.
I can make a note of that,
that
in
is
a
full.
moment.
Okay, but let's write down 6 and 7 first.
I don't know what this is.
Yes, yeah.
Steve said it was some sort of tunica.
Tunicate?
Thank you, Nav.
Yeah,
Is
this
that
is
something at 8 inside, or?
No, that's part of its anatomy.
Okay.
Sometimes they have polycate worms inside, polynodes. This is a tunicate You can kind of think of it as like similar to the flytrap anemone. You know, some prey will climb in there and it'll close its mouth, which is on the
250.
downward side, and consume it. The genus is Megalodicopia.
Say that one more time.
Megalodicopia.
Megalodicopia. That's a cool genus name. I like it.
All right,
coming out.
nice. We've seen a few of these so far on this expedition.
It's kind of a big one.
Yeah.
Very